Welcome. This tutorial here is for the classic dominance Mendelian genetics for the monohybrid crosses. I have regular paper here. I think it's easier to explain it as I'm writing things instead of you just watching me typing on a screen. So you should be working on a screen, or if you want to write it out, go ahead, print it out, and let's get into it. All right. So in dogs, the gene for fur color has two alleles. The dominant allele, capital F, for fur, codes for gray fur, and the recessive allele, little f, codes for black fur. So right off the bat, it's telling us what is dominant, what is recessive, and which letters to use. So we don't have to even think about much of anything. It gives us letters, we don't have to come up with letters. It tells us what's dominant, what's recessive. Great. All right, so number one, the female dog is heterozygous. The male dog is homozygous recessive. Figure out the percentage ratio of the possible phenotypes of genotypes of the puppies using the Punnett Square. So break this down bit by bit here. So I'm also using different colors, which gets me very excited. So right off the bat here, for our female dog is heterozygous. So that tells us right off the bat, hetero means different. She'll have different capitalizations for her alleles. So in our little first table here for 1A, we can fill out her genotype. We know she is heterozygous. She's going to have one of each allele. So that means she's going to have a capital F and a little f. And since we know the dominant allele F codes for gray fur, if we see a dominant allele in classic Mendelian dominance, if you see that dominant allele, your phenotype is going to be the dominant phenotype. So she will be gray. All right, now let's look at the male dog here. The male dog is homozygous recessive. Great. Homozygous means we're going to have the same capitalization. Recessive tells us if it's going to be upper or lowercase. Recessive will be the little one. Recessive is always going to be the lowercase letter. And he's homozygous, meaning the same capitalization. So we'll have a little f and a little f. Now, when we have homozygous recessiveness in classic Mendelian genetics, that means they will be the recessive phenotype. Recessive phenotype. And in this case, the recessive allele codes for black fur. So our male here will have black fur. Awesome, awesome. Now moving up a little bit, we're going to need to make a Punnett square. Best practice says male goes across the top, females go down the side. You can kind of do whatever you want, but I'm going to try to make the best example I can here. So we already know our genotypes. We just got to plug them into the outsides, the top and the side, and then we can fill in the rest of our Punnett square to find our offspring. So first up across the top, we're going to have the male. And we know his genotype is FF, so we're going to put one of each of these alleles into our Punnett square here. One allele per column. Great. Now, our female, I can write sideways. There we go. We also have her genotype ready to go from letter A. We're going to, just like the male, we're going to put one allele per row instead of column. Start with this big F. She also has a small f. There we go. We figured out the parents. Those are plugged into our Punnett square. Easy peasy. Now comes the filling in. These four squares represent the potential offspring between these two parents. All right, so from the female, we're getting big F's across this way and little f's across this way. Best practices tell us capital letters always come before lowercase letters in our genotypes. So I'm going to go ahead and write these capital F's because they're going to come first. And then it doesn't matter which one comes first here because the male is only going to give us little f's. So I'll just keep consistent with my colors here, do our two little f's for the female. Great, so our female donated one allele to each of her offspring, Now the male has to come in and give his single allele to each of the offspring. Little f there, little f there, little f there, little f there. Awesome, awesome. Great, great, great. So now we see that the female gave one allele for this trait. The male gave one allele for this trait. So our puppies now have two alleles. Right on. That is what we want. Without those two alleles, we won't know what the genotype or phenotype will be of our puppies. Next step, letter C. What is the genotypic ratio of our puppies? All right, I'm going to have to break out some other colors here. Let's do some green. All right, so for green, genotypes, what letter combination capitalizations are we looking at here? Focus. All right, so in these top two, we have one capital and one lowercase. So we have one capital here, one capital there, and let's use orange, and one lowercase, one lowercase. And we have two of those. So we can say we have two of the four or 50%. Our heterozygous big F, little f. Now looking at the last two here in the bottom, our last two puppies, these are all little f's. So they're just all going to get these orange lines underneath them there because the orange represents our little f's. They are homozygous recessive. Homozygous recessive. Homo means the same. They are the same capitalization, and it's recessive because those are the little f's. No capital F's in these bottom two puppies. All right, so now we took a look. I'm actually going to make these into boxes. The lines aren't very easily seen here. All right, that's a little bit helpful. I'm going to use my green over here to there we go. All right, so now across the top we have two that are heterozygous, big F, little f. And across the bottom, we have another two that are little f, little f. So what does our genotypic ratio look like? Well, do we have any of these? Let me change my color again. Do we have any of these big F, big Fs? We don't. So there will be 0 of those. Awesome. Now we have two of our heterozygotes. So this is big F, little f. And we have two big F, little f's. And lastly here are little f, little f. We have two of those as well. Awesome, awesome. So this blank, this long blank here is to help us write out our final ratio. So there's a couple ways you could do this. I prefer to use percentages because a lot of the time that makes sense to me, but sometimes I also prefer using number ratios. It all depends on what you prefer, the various problem you're on. Just kind of go with it, I guess you could say. So I'll do both of them just so that you have both options available should you want a preference. All right, so we know we have 0 of these homozygous dominant, the big F, so we don't have to worry about those. Now we have 2 of our heterozygous and 2 of our homozygous recessive. So of the 4 total, I can also write this, of the 4, we're either going to say we have 50% big F, little f, 2, this colon usually means 2, 50% little f, little f. The other way you could write it if you don't like percentages is you could say 2 heterozygous, 2 homozygous recessive. It's really important that you make sure you write these sort of descriptions of what that ratio is. Otherwise, it could be what? If I don't have these here, it could be 2 that are homozygous dominant. We don't know. That's why we have to be specific and make sure we write the little description here of what genotype we're talking about. All right, now let's move on to D, the last little part here for our puppies. What is the phenotypic ratio of the puppies? So now we're looking at the phenotype. Change colors. Let's get out some highlighters in a moment. So we're looking at what are the colors of the puppy's hairs going to be. All right, so let's go back up to our Punnett Square really quick. We know that gray is dominant, black is recessive. So knowing that, up here on our heterozygous, our 50%, 2 of the 4 heterozygous puppies, they have that dominant allele of big F. That means they're going to express or show that dominant trait of gray fur. Let's use actually not that one, green highlighter here. So for the phenotype, I'm going to color in with my highlighter. So half of our puppies have that big F, they will be dominant. The other 2 puppies, they are little f, meaning recessive. They don't have that dominant allele to sort of take over, so they will be that recessive black color. For this one, I guess I will use my orange marker since I already have orange going there. Orange highlighter. Awesome, awesome. So now we've highlighted to indicate our phenotypes. Cool, cool, cool. Keep this there. All right. So what is our phenotypic ratios? How many of our puppies are black? Well, looking here, black is recessive. We have 2 of the 4. And how many are gray? 2 of the 4. Now in this long box, we can write our actual ratios instead of just counting. So again, we can start off with percentages. I like percentages when we're doing these sort of more basic monohybrids. So 2 of the 4 gives us 50%. Gray. Remember, we need our little descriptors there. 2, oh, sorry, this is supposed to be black. Oh, no. Black, 2. You could have just kept gray, but I like to go in the order of the information I'm given. 50%. Gray. Now, if you like just the numbers, no problem. Let's do those. We have 2 that are black. So 2 that are gray. So there we have it. We finished problem number 1 with our puppies of various coat colors, fur colors. All right, let's move on to number 2 here. Getting to the end of my paper. I don't think yours is quite there yet. So in dogs, hereditary deafness is caused by a recessive gene, little d. So I'm going to take a moment. I will assign the blue color. So recessive gene, little d. All right. Now a kennel owner has a male dog, Gilbert, that she wants to use for breeding purposes if possible. The dog can hear. So again, the deafness is recessive with the little d. So what are the 2 possible genotypes of Gilbert? How do we know he has 2? Well, he has that dominant trait of being hearing. He can hear things. He's not deaf. So what are the 2 possibilities for Gilbert's genotype, the letters that represent his alleles? Well, if hearing is dominant, we're going to need that capital letter to signify our dominant trait. And if he's homozygous dominant, homozygous means capital, capital, and dominant because they're big, they're tall, they're capital Ds. Now, there's another possibility in classic Mendelian genetics. He could also be heterozygous. That's because if there's at least one dominant allele, that dominant trait will be expressed. All right, So that means he needs at least one dominant allele. And if he's heterozygous, he's still going to be hearing, but he will have just that little heterozygous mini recessive gene that is not being expressed. All right, let's move on down. All right, so if the dog's genotype is in fact DD, we are not sure. We're just going to try to play pretend here, give it a shot. The owner does not wish to use him for breeding so that the deafness gene will not be passed on. That makes sense. It's kind of a good thing to have a dog that can hear. So we don't want to pass on that deafness gene so that our dogs can hear. This can be tested by breeding the dog to a deaf female, little d, little d. So I'm going to put another box around that to call attention. Fill in these 2 Punnett, oh, whoops, I should have given you 2 Punnett squares. I will do that on yours so that it won't show up here as I'm doing it, but on the one on the doc I share, you will have 2 Punnett squares. Fill in these 2 Punnett squares to illustrate the crosses of your possible male genotypes from question. Again, I'm going to fix this, finding these typos, from question A with the female deaf dog. All right, so we know what the female's genotype is. She's deaf, and deaf is recessive. That means she is going to be little d, little d. We figured that out from the question here. Best practice says females go down the side, males goes across the top. So little d for the female. And we need to figure out what Gilbert is. Is he homozygous dominant or is he heterozygous for deafness? Okie doke. Well, we're going to do 2 Punnett squares anyway. So let's just start off with the homozygous one. We're going to do a big D, big D. All right. So now, actually, I'm going to make this purple. Let's give a little bit of contrast here. Big D, big D. All right. Best practices tell us capital letters come first. So I'm just going to drop down some D's here just to get them out of the way. I know they'll come first. All of these puppies are going to get a capital D from Dad. And going across, we're going to fill in all of these little D's from Mom. Cool, cool, cool. Let's do our second Punnett square really quick so that we can figure out Gilbert's genotype here. All right. So again, we know what the female is. She's going to be little d, little d. And the other option is this heterozygous big D, little d. Now, we know big D is going to have to come first for these puppies here. So big D, big D. And then it doesn't really matter because all that's left are these little d's. So just because I had the purple pen in my hand, I'm just going to do those little d's first. This is little d, this is little d, this one's little d, this one's a little d. Okie doke. So now we have our 2 possible father genotypes. What are the genotypic ratios of the puppies? So once again, I'm going to cut these in half for our 2 Punnett squares. You on yours will have 2 separate ones of these. Right. All right. So there we go. What is the genotypic ratio of the puppies? How many are big D, big D? None of them. I don't see any double purple, double big Ds. All right. So 0 big D, big D in both of these Punnett squares. Let's say this is Punnett square 1, this is Punnett square 2. This is 2, 1, 2, 1, 2. All right. Next up, how many heterozygous? Big D, little d. Interesting. So in Punnett square 1, 1, 2, 3, 4 of them are heterozygous. Interesting. All right, so 4 of the 4. How about Punnett square 2? We have These two right here are heterozygous. There's a big D and a little d. So we have 2 for Punnett Square 2. Now, what about these little d's? Well, in Punnett Square 1, we found out all 4 of them were heterozygous, so we know there's not going to be any there. So we're looking for more little d. Look at that. We got 2 of those there. Great. Now we're going to have to write it out. So for Punnett Square 1, again, we'll do 1, Punnett Square 2. All right. What is our genotypic ratio? Well, 100% of them, or 4 out of 4, were this heterozygous big D, little d. So we can do 100% big D, little d, or we can also do our numbers. You can say 4 to 0, because we don't have any other genotypes, big D, little d. Now let's look at Punnett Square 2. Punnett Square 2, we had no big D. We had 2 big D, little d, 2 heterozygotes, and 2 little d, homozygous recessive. So that means we had 50%, or 2 of the 4, were heterozygous to 50% homozygous recessive. Or, if you like the numbers, we'll do 2 big D, little d, to 2 little d, little d. All right, moving on. What is the phenotypic ratio of our puppies? Okie doke, let's get out our highlighters one more time so we can visualize here. This time I'll do pink and blue, pink and yellow. I like yellow. Let's do pink and yellow. All right. So for this one, all of them are going to be heterozygous. Great. They're all this phenotype. They're going to display that dominant trait because there's at least one dominant allele. So for Punnett Square number 1, got my black line here. What is the phenotypic ratio of the hearing puppies? Well, we have 4. Again, let's see this 1 and 2. We have 4 that are hearing. Awesome, awesome. Next up, let's look at Punnett Square 2. We have some differences here. We have 2 with that dominant allele, that capital D, but the other 2 are homozygous recessive. They will be deaf. They have that recessive gene for deafness. So that means, oops, 0 that are deaf. Looking here, we have only 2 that are hearing. They have that dominant trait of hearing. And we have 2 that are deaf. All right, we got our long box here to sort of write out our ratios. We're gonna have for number Punnett Square 1 versus Punnett Square 2, 100% will be hearing, or 4 to 0 hearing. For Punnett Square 2, we got a 2 and 2 here. So 50%, or 2 of the 4, will be hearing, to 50% deaf. If you like the numbers, we'll do 2. I'll do H for hearing, to 2 that are deaf. Sorry, I'm in another room. All right, now explain how we can tell the genotype of Gilbert. Interesting. So I'm going to run out of room. That box is not big enough for me to write it. How can we tell the genotype of Gilbert? Well, based on what the puppies give us up here in our Punnett squares, if Gilbert is capital D, capital E, homozygous dominant, all of the puppies he makes with the deaf female will be hearing. So if the breeder gets, let's see, if there are no deaf puppies, Gilbert is homozygous dominant for hearing. Well, what if he is this heterozygous? Look at the puppies. If Gilbert is heterozygous, half of his puppies will be hearing, which we figured out from our little phenotypic ratio here, and half of the puppies will be deaf. If half the puppies are hearing and half are deaf, Gilbert is hetero. Whoops, I can spell Gus. Sorry, my handwriting is rushed right now. So that's how we can figure out what Gilbert's genotype will be. If there are no deaf puppies, Gilbert is homozygous dominant. So that means big D, big D. And if half the puppies are hearing and half are deaf, Gilbert is heterozygous, meaning big D, little d. All right, that will be the end of our regular Mendelian classic inheritance.